Good afternoon, and thank you for the opportunity to hear a little more about OS Therapies. We've had a very busy 2020, and we're looking forward to a very exciting 2021. My name is Paul Romness. I'm the President and CEO of OS Therapies. Joining me on this presentation today is Dr. Robert Pedrick, the Chief Medical and Scientific Officer for OS Therapies, but also really the father of our original platform technology, the OST HER2 platform technology. As you see in the pipeline slide in front of you, we started OS Therapies originally around osteosarcoma. We have now, in 2020, expanded to all other indications in the human for this technology, as well as the already provisionally approved canine indication for osteosarcoma. Also this year, in 2020, we added another pipeline platform technology, what we believe to be the next generation antibody drug conjugate with proprietary Cy-link or silicone linkers that are really what we think is going to be the special sauce in that technology. We're really talking about a great opportunity to invest in OS Therapies right now. We've just started finishing up our $5 million A round, and we're about to kick off a $20 million B round. That will pay for our clinical trial that you see on the slide. We have a phase 2B registration trial that we believe will have an FDA approval in the next 24 months. That will also provide us with a priority review voucher, which has a significant value to it. We've also recently added the canine indication, and we are looking for a global partner to share that and to commercialize that. And we are also doing preclinical and toxicology trials on our new platform technology, the tunable drug conjugates, or the next generation antibody drug conjugates. It is a very hot market, the ADC market. I think you'll see in some of the comps later a great opportunity to not only develop new therapeutics with this platform technology in addition to solid tumors, but also great opportunities to out-license some of the technology to other larger pharmaceutical companies. We'll also talk about the finances, what we're doing from a go public strategy as we talk this today, and just the opportunity for you to invest in what we think is not only a do good, but a do well investment. Robert, I'd love if you could talk about our two platform technologies on a very high level and how they interact with each other, and then we'll get into the details of those a little further.
Absolutely, Paul. So these are very modern technologies that are taking advantage of some of the latest advances of what's known to be effective in cancer treatment. Our lead product is an immunotherapy that's based on a fascinating genetically engineered bacteria that delivers cancer antigen targets and at the same time stimulates the innate and adaptive immune system, making the immune system receptible for receiving new messages and allowing it to see the cancer where maybe it was ignoring the cancer before through immunostimulation. This is a fascinating treatment because it can be given over and over again, and each time it stimulates a new round of immunity, which really generates armies of white blood cells that have the ability to find where cancer cells may be hiding, micrometastases, surround them, kill them, and eliminate them so that the cancer doesn't come back and that the patient goes on for a prolonged survival and doesn't have to deal with the cancer coming back. Fascinating. We're able to engineer into this the ability to put different payloads here, and the payload we've chosen is HER2. We've all heard about HER2 positive cancers, and this really gives this a hold on the osteosarcoma, but it also opens up the opportunities for this product in other cancers like breast cancer, esophageal cancer, and other HER2 expressing malignancies. Fascinating technology, and it's already been approved provisionally for treatment of canine osteosarcoma, and where it works very, very well, and we'll talk about that a little more. Behind that is another modified composite product. These are our tunable antibody drug conjugates. The antibodies are the targeting portion, and these antibodies are trained at a cancer target, and they do what antibodies are designed to do. They find, they circulate through the bloodstream, and when they find the target, in this case the cancer cell, it sticks to it. But these antibodies are not just plain antibodies, they're loaded with multiple copies of a toxic payload. The cancer cell internalizes these antibodies when they bind to it, and once inside the cancer cell, these payloads get released, and they're like little hand grenades. And basically, the cancer cell just swallows these hand grenades. And with the tunable drug conjugates, we're able to control how many payloads, how many hand grenades they have. In the case of our lead product here, they're packing 6 hand grenades. And we also can control how it's released inside the cancer cell. And that's really taking this technology to the next level beyond what first-generation antibody drug conjugates can do. Our initial target there is going to focus on folate receptor alpha, which is very highly expressed in some gynecologic malignancies like ovarian cancer and also some other things like endometrial cancers that we'll be looking at. This is at the preclinical stage, but just as soon as we push through and get the osteosarcoma study sort of in the bag and about ready to finish, these will be hitting the clinic with our first conjugate of our tunable drug conjugates. So this is an opportunity for a near stage product that's one trial from potential registration, and it's a platform that provides the potential for multiple products, which also provides multiple licensing opportunities based on the tunable drug conjugates following it up. Thank you.
And I'll brag on Robert and his team. They've really done a great job. They initiated their weekly clinical trial meetings right as COVID was beginning, and they have a protocol that's ready to go, and it looks like we're going to start our phase 2B this quarter. It's 39 patients, and we'll talk about that a little bit more. It's really exciting because we are very confident now about a priority review voucher. We already have orphan drug status, and we also already have fast track, and we believe that we will get breakthrough technology designation on that technology. Just a little bit about our management team. I've been around a long time. Robert's been around a long time. Luckily, we have Dr. Uta Vanna, who is the technology expert behind the tunable drug conjugate. So in Uta and in Robert, we have the scientists that developed this technology. And I know for a fact they want to get this into the clinical trials and show all of you, and show the FDA that this works. Our board of directors is ever expanding. We will soon have Dr. Colin Goddard, the CEO of Blink Bio, as our executive chairman of our board. He is joined by John Ciccio and Ted Search, both presidents and CEOs of very successful healthcare companies, as well as Olivia Eggy, who is a osteosarcoma survivor. And she is an emerita member of the board, but she gives us the patient perspective, which is very important to us. Our scientific advisory board is very heavily osteosarcoma focused at this point. That will evolve. These are the who's who's of osteosarcoma. The only two that probably aren't on here is our principal investigator, Dr. Damon Reed at Moffitt, or Dr. Katie Janeway, who is the chairman of the Bone Committee of the Children's Oncology Group, which we'll talk about here in a minute, and she's at Harvard and at Dana-Farber. That scientific advisory board will expand. We also wanted to share with you the market potential on osteosarcoma. We believe that if this technology works in osteosarcoma, and these are very conservative numbers, that we will pivot to other much more prevalent solid tumors such as breast cancer and esophageal cancer. But we believe that we can make a significant impact on our bottom line by this indication alone. And our 5 to 7 year financial plans are very robust and include not only osteosarcoma and the other indications for the OST HER2 platform, but also the ever-advancing tunable drug conjugate OST TDC. We talked about the priority review voucher earlier. If our technology works as well as we believe it does, and it did in dogs, we will get approval from the FDA. That gives us a transferable voucher, which larger pharmaceutical companies who have large blockbuster products buy from smaller pharmaceutical companies, biopharmaceutical companies like us, to cut in line at the FDA. And it's usually a 3 or 4 month advantage for them to get to market earlier, to start their growth in their overall sales of that product line, and to beat the competition to the market. So here's a great opportunity to have some non-diluted funds coming into the company in around 2022. An overview of the OST HER2. Robert, you're much more of an expert on this. I can do it, but I enjoy your perspective better.
Sure. So this is a fascinating product, as I said. It is essentially a preventative cancer therapeutic vaccine, so to speak, not something that's given to prevent an infection, but once the patient has cancer, this can be given to stimulate the immune system to better be able to find and eliminate the cancer. This is specifically interesting because this vaccine generates T cells. T cells are what everyone is focused on now in the immunotherapy of cancer, because we know that the T cells are the ones that kill the cancer cells in hand-to-hand or cell-to-cell combat, so to speak. This is a trial that's going to be done on an active IND. Sorry, could we go back one? Thank you. So we've already had a phase 1. This, as we mentioned, the fusion protein here that's been delivered by this engineered bacteria is HER2 specific. This was originally developed at the University of Pennsylvania, which has emerged as a focus of cutting-edge immunotherapies. And we have a patent coverage that covers us all the way out to 2031 with this particular composition of matter. This is not a new product in man. There's already a phase 1 that's been conducted, a safety study in adults with various cancers. And we are also have the benefit of this is of being able to develop in canine osteosarcoma at the same time. Right now, there's an ongoing phase 3 trial with 180 patients in canine osteosarcoma with this agent, which at an interim analysis has already shown a tremendous improvement in survival for the canines, which with a highly statistical significant level here. And so we're looking forward to, when that data is made publicly available, having a huge emphasis on the canine indication for this disease. But as Paul said, canines are great. We love them. They're our four-legged family members, but we want this available for the children as well. So we're ready, almost ready to start our phase 2 trial in partnership with the Children's Oncology Group in pediatric osteosarcoma. Now, the Children's Oncology Group is the people all over the country that are affiliated with the government and the network that focus primarily or solely on children's cancers, and that's where almost every patient with osteosarcoma goes. So those are the key people, and they're so excited about this, they've actually partnered with us in some way.
And why is it so important that we develop this technology? In osteosarcoma, as well as in breast cancer, the survival rate significantly decreases when metastases come into the lungs and the brain. If you think of friends and family members that have had breast cancer, it's very much like osteosarcoma. It's a solid tumor that gets resected. We blast the body with chemo and radiation, and then these little micrometastases, what we call cancer seeds, that survive. That chemo radiation regimen in osteosarcoma is the worst in cancer. Cisplatin, doxorubicin, and methotrexate. Any of those could kill a grown man or woman. And here are teenagers taking this stuff. And unfortunately, if it does come back, they've got about a 13% chance of survival. So we believe we have a solution for this, and that's why we're going to do the trial. You're talking about how the micrometastases are attacked, Robert. This is actually a slide from the phase 3 trial where the lymphocytes are attacking the micrometastases or cancer seeds, if you'd like to get in more detail about that.
Yeah, absolutely. So once a patient has received their chemotherapy and radiation, in many patients, half the patients with osteosarcoma, they are effectively cured. The other half, they were almost cured. But there are some survivors, little splinters of the cancer that break off and set up housekeeping somewhere else in the lungs or in the brain. We call them micrometastases. Eventually, these will continue to grow and get big enough to where they can be detected clinically and cause a recurrence of the cancer, which ultimately is what what becomes fatal for most people with osteosarcoma. Wouldn't it be great if we can find where these cancer seeds are hiding inside the patient and send a different technology, not chemotherapy, not radiation, something that can find every individual cell and determine if it's good or bad, and surround it and kill it. And that's exactly what we're doing here. And this is literally a picture of osteosarcoma cells in a micrometastasis in a lung of a canine that are surrounded by these darker cells, that are T cells that are fighting against it. This particular dog never went on to get any more cancer after treatment. So that's exactly what we're trying to do in these patients. Take out the cancer when it's at its weakest, as little single 1-cell, 2-cell, 3-cell micrometastases using the immune system.
And I think we already covered this, but we have an active IND. We're ready to start the trial. Fast track and orphan designation. We believe that we'll get breakthrough designation as well as the priority review voucher. We anticipate a meeting with the FDA, but if we don't, we're going to start the trial without it because we have the IND. If we do get a request from the FDA to come and visit them, we'll have a host of folks that want to meet with them that are very excited to support what we're doing. The FDA is also very excited about what we're doing as they've been briefed about this clinical trial many, many times. The trial is 39 patients. We believe we will be able to enroll it in 3 to 4 months, which means that we would be filing with the FDA less than 2 years once we start the trial. The two primary endpoints are overall survival and disease progression. We know those to be reliable endpoints for this trial and for this disease because this is what was used for the preliminary approval or the approval for the canine indication. This slide, and Robert, you can talk more of the details of the genetics of it, but this is showing that osteosarcoma in humans and in canines is almost genetically identical, and that's why we believe this technology will work in humans as well as dogs.
That's correct, Paul. Even a genetic analysis of gene expression in the two cancers shows that it's the same mutations that drive the cancer, and it's the same mechanism of action that causes the cancer to initiate in the bones and spread to the lungs. Here we see human bones and lungs on the left, canine bones and lungs on the right. It's the same disease, and so since this works so well in the canine version of the disease, we're very encouraged that it may also have the same effects when we are able to demonstrate that in our pediatric patients.
So these are some of the clinical results that show the 3x improvement in overall survival versus standard of care. Another proof of concept phase, this is the results of the phase 1 canine. As Robert mentioned to you, there is a phase 3 trial that we know that the p-value is 0.007 between the treated and untreated arms. That is, for any of you statistics experts, is significantly significant, right? Statistically significant.
Highly significant.
Highly significant, right. So turning to our newer and I think as promising or even in the long term more promising platform technology, the tunable drug conjugates. This is a really exciting technology. I think most of you, if you've invested in them or if you've been following the space, you know that there have been some really big deals in the antibody drug conjugate. We believe this to be the next generation of that because of the silicone linkers that are affected by pH that gives us the ability to drop payloads specifically into cancer environments. I wanted to get to the comps on some of the deals being done in the ADC space. If you followed Immunomedics, they were just recently bought out by Gilead for the tune of $21 billion, I think it was. Merck has been putting more and more money into Seattle genetics. There's just a lot of excitement in the antibody drug space right now. So we're taking advantage of that, and we believe that there's going to be an opportunity for us to outlicense much of our technology. We also have very strong patent protection through 2037 and 2036, particularly on the linkers. Ironically, in all other ADCs, the weak link is the linkers, and I'll let Robert explain that.
So an antibody drug conjugate is just what it sounds like. There's an antibody that's the targeting moiety, and then there are payloads, or I described them earlier as hand grenades that are attached. The challenge with antibody drug conjugates in the past has been making sure that the hand grenades don't go off in the wrong place and cause bystander damage to normal tissue, and making sure that they get a lot of them inside the cancer cell itself. This particular chemistry with the Silinker is proprietary to Blink Bio and fascinating, and perhaps the solution that has that will help enable these drugs to be more widely utilized going forward. We are able to put 6 different payloads on here, and we call them tunable drug conjugates because we can tune, based on the chemistry of the Silinkers, we can tune exactly how the payload will be released and in what conditions. And understanding what the target cell, cancer cell, is like on the inside, we can make certain that those payloads get released inside the cell. This is a cartoon which shows that just exactly what I'm describing here. The payload binds, or the antibody binds to the cell, where the whole complex is taken into the cell in a process called endocytosis. It gets inside the cell. Once inside the cell dumps acid onto those chemicals to try to break them down. And what happens is that releases the payload, the hand grenades. And so now with each antibody that binds to a cancer cell carries 6 molecules that are all very highly toxic. We're going to be using known cytotoxic chemotherapies in the beginning, but this technology is so versatile we can also attach biological targets and even nucleic acid targets to be delivered specifically. So we have a lot of excitement about our first product. It's going to be focusing on folate receptor alpha, and here's some of the preclinical data showing how highly effective those are at killing the cancer and curing the mice of their cancer xenografts, and that they're cleared rapidly, and that they're not toxic because they have these cap proteins on them. So our first target payload or hand grenade is going to be a very potent chemotherapy agent known as exatecan. Exatecan is of interest because it's highly active, but it's been very difficult for other people to attach to an antibody drug conjugate because they don't have the Silinker technology. Our Silinker technology allows us to attach multiple copies of hexatecan to our antibody drug conjugates that are going to be targeting the folate receptor initially, which is overexpressed in a large number of cancers, particularly gynecologic cancers, which we mentioned. These are some of the cancers that we mentioned, ovarian and endometrial. You can see it, but this is also overexpressed in about 74% of adenocarcinomas of the lung. So that really gives us a lot of potential, even for our first antibody drug conjugate, to have a very significant market.
And I think we've covered most of this around the opportunities and advantages for the TDC. The really exciting thing is that silicone linker is affected by the pH level in the cancer environment, and we can affect that solubility of that linker depending on the pH of the environment we want that payload to be launched at or dropped in. Again, we have great coverage for that from an IP standpoint globally, just like we do in the HER2 technology. I think we shared our strategic plan earlier, but I want to show you that we really are ticking off things that are important and moving through. We're successful with the addition of the pipeline, and we are now discussing a go public strategy, which includes a listing on the OTC, where we'll be going public in the next 90 to 100 days. That's part of our $20 million B round that you see here. The timing on these is still very accurate. We're just finishing up the $5 million A round. We're about to start the study. If the FDA wants to meet with us, we will meet with them. We believe we'll have full clinical trial enrollment by Q3 of 2021, which means that we would be able to get some breakthrough designation in the same time frame next year with some of the interim data, and then look to a approval from the FDA in first quarter of 2023. For those of you that follow drug discovery, this is wickedly fast. The reason the FDA has approved many drugs in recent times with only a phase 1 or phase 2 trial because the results of the disease otherwise are so bad. In the case of osteosarcoma, that's the case as well. Our costs are very much kept under control, not only by us, but by our vendors. Our vendors are partners with us, including the Children's Oncology Group and George Clinical doing our CRO work. We're very transparent about our budget. We are ever expanding that budget with the new technology, and we look forward to you partnering with us. There are nonprofits who are also partnering with us who have taken equity positions or not. If you know the Cystic Fibrosis Foundation model, that's what we're trying to emulate here. And we really look forward to having you join us. As we started this fight a long time ago, 3 years ago, I said, we all do what we can do. I hope you can help us. Thank you.
Thank you for this presentation. It's a very powerful platform. You're actually combining cancer vaccines with TDCs. ADCs have been such an attractive target nowadays. So I am curious. I would like to learn about the osteosarcoma data. When would you disclose? I know you will be initiating shortly. You'll complete the enrollment. Do you plan to disclose data? And if so, what does the timeline look like in your mind?
Robert?
So if enrollment goes as we think it will, because everyone in the osteosarcoma community is excited about this trial, and as Paul mentioned, we think the trial will enroll in about 4 months. It takes about a year to complete treatment, so the last patient a year after, we'll start the trial probably this quarter. As soon as we can, add 4 to 6 months for enrollment, and a year after that, the trial will be complete, and we'll be able to disclose that data and be preparing our filing. So I think that works out to what, third or fourth quarter 2022, right, Paul?
Yeah, but we'll get a cut on the data in early 2022 for the breakthrough designation filing.
Mm-hmm.
So you wait for the completion of the enrollment to disclose any type of data.
Right, and the primary endpoint in the trial is looking at the proportion of patients that have had a recurrence or not of their osteosarcoma at a year's time after they've started treatment. Because we know historically 80% of those patients will have a recurrence within a year without treatment. And we're going into a space where there's currently nothing to do but to watch and hope that the cancer doesn't come back. And so there's not really a need for a control. The historical data on this goes back 30 years. They know exactly what it is. So.
Not only that, but I would love to say, hey, we were brilliant in our strategy of starting with osteosarcoma because we had an opportunity for a priority review. We could do the trial quickly. We could see if it works well in a solid tumor. When we started OS Therapies, it was the only indication we had. Now we have all the indications. We have the canine. Now we have the ADC platform. So we have many more shots on goal. But if we had it all over to do again, we would do it in this sequencing, because the PRV will fund us, as well as some of the licensing opportunities around the ADCs will fund us before we get to market.
That's understandable. I'm also curious, I cover immunogen and also advaxis previously, but the TDC platform, what is the reason you are going after, especially for folate receptors? Was there a reason behind it? What is the scientific rationale behind it? And also, what are you looking at for the next steps? Are you planning to expand to other indications, since it's a platform technology that can be utilized in many other directions?
Yeah, well, I think that initially it has to do with the fact that, in my opinion, gynecologic cancers are underrepresented in the treatment of cancer. Immunotherapies have not been largely effective, and they need better treatments there because it's a significant disease. The folate receptor is very highly expressed, in particular by ovarian and cancer and other gynecologic malignancies. And the problem was that no one had the chemistry before without the Cy linkers to be able to go after the folate receptor with something like exatecan. We have that technology here, and so it's an unmet need essentially in gynecologic cancer, and it will give us a foothold and provide an effective treatment, we hope, in a disease that is in desperate need of some new treatments based on more modern technology than the old chemotherapy. So we're excited about that. But as we described, this target also has activity in There's also expressed in lung cancer. Well, there's a lot going on in lung cancer, and that will be an interesting development pathway. But right now, we think that it's a hugely unmet medical need, and because of the Cy linkers and our ability to get to the exatecan with this technology, that makes perfect sense for us to go forward.
That's understandable. So I have one more question. I don't mean to be repetitive, but I'm really curious when you would get the first human data from the TDC platform and any clinical data. What does the timeline look like on that one?
So on TDC data, we are actively initiating our preclinicals and our toxicology 2-week and GLP tox. We hope to have those done in the next 6 to 8 months. That would then give us the ability to apply for the IND through the FDA, which would mean that we would be able to get started on a trial with those TDCs in early, if not first quarter of 2022. But once we get through the GLP tox, we'd have the ability to out-license the Cy linker technology to some of the companies that we know have had some challenges with their linkers.
That sounds great. Actually, it sounds like you have such an exciting 12 to 18 months coming up. So looking forward to following the story. Thanks for your participation.
Great.
Thank you.
Thank you.